Hi, my name is Ali Gifford, and in this video I will introduce you to the Algonauts Project 2023 challenge, called How the Human Brain Makes Sense of Natural Scenes. I will start by introducing what the Algonauts Project is. Next, I will move to this year's edition of the project, the 2023 challenge, and tell you about its goal, the data on which it is based, and how we will evaluate your submissions. Finally, I will walk you through the associated Colab tutorial, which is aimed at familiarizing you with data loading and visualization, modeling, and submission to the challenge website. Since its inception in 2019 and later in the 2021 release, the Algonauts Project is a platform that promotes interdisciplinary interaction between biological and artificial intelligence researchers. The project's core is an open challenge with a quantitative benchmark, where participants compete in accounting for brain activity through computational models and algorithms. The project aims at providing better models of intelligence to rapidly advance scientific discovery in both biological and artificial intelligence fields. The goal of the 2023 edition of the challenge is to prompt the fields of computational neuroscience and computer science to come together towards building cutting-edge encoding models of the visual brain. In this challenge, encoding models consist of algorithms that take images as input and predict the corresponding brain's visual responses. The 2023 challenge gives you the unmatched opportunity of building neural encoding models using the most massive dataset of human brain responses to visual scenes, the Natural Scenes Dataset, in short NSD. NSD is a large-scale fMRI dataset collected at the University of Minnesota, and it provides high-quality fMRI responses to 73 different stimuli images across 8 subjects, where all the stimuli images consisted of naturalistic color scenes coming from the COCO database. The Algonauts 2023 challenge uses pre-processed fMRI responses of all 8 NSD subjects that have been projected onto a common cortical surface template. Brain surfaces are composed of vertices, and the challenge data consists of a subset of cortical surface vertices in the visual cortex that were maximally responsive to visual stimulation. Here you can see these vertices shown in purple. We provide the data in right and left brain hemispheres, each consisting of different amounts of vertices. During the challenge, you're invited to build computational models that predict these vertices' responses to images. Since model building requires independent data splits for training and testing, we partition the challenge data into non-overlapping train and test splits. The train split comes from the publicly released NSD data, and it consists of 8,000 to 9,000 unique images per subject, along with the corresponding fMRI visual responses. You can use this data to train your encoding models. The test split comes from the last 3 NSD scan sessions, which are not publicly available, and it consists of 200 to 300 unique images per subject. However, we withhold the corresponding fMRI visual responses, and you can use your encoding models to predict the brain activity of these test images. The visual cortex is divided into multiple areas, having different functional properties, referred to here as regions of interest, ROIs. Along with the fMRI data, we provide you with indices for selecting vertices belonging to specific visual ROIs. Here you can see the surface plots of subjects 1 ROIs, where each ROI is color-coded, and the names of missing ROIs are left in black, since not all ROIs exist for all subjects and hemispheres. However, keep in mind that the challenge evaluation metric is computed over all available vertices and not over any single ROI. To quantify the accuracy of your encoding model, you will submit its fMRI predictions for the test images. We will then compare these predictions against the withheld fMRI responses to the same test images collected during the NSD experiment. We will perform this comparison by correlating your fMRI predicted data with the corresponding ground truth withheld data at all vertices of all subjects across the test images. And the overall challenge evaluation metric, which quantifies the performance of your submission, is the median noise normalized square correlation score over the vertices of all subjects. This metric reflects the percentage of predictable variance accounted for by your model, given the noise in the data. For more information on the Algonauts Project 2023 challenge, please see the challenge website and paper by scanning these QR codes or by following the links provided in this video's description. Along with the challenge data, we also provide a Colab tutorial in Python where we show you how to load and visualize the fMRI data, its ROIs and the corresponding stimulus images, build and evaluate linear Bayesian encoding models using a pre-trained AlexNet architecture, and prepare predicted brain responses of the test images in the correct format for challenge submission. In the remaining part of this video, I will walk you through this Colab tutorial. Now I will walk you through the Colab tutorial of the Algonauts 2023 project challenge. And in this tutorial, you will learn how to load and visualize the challenge data, how to use this data to build encoding models that predict fMRI responses to images, and then how to prepare these predictions, and specifically the predictions of the test images, in the right format for submission to the challenge leaderboard. You can run this tutorial here on Google Colab, or if you prefer, you can also run it locally on your computer via Jupyter Notebook. And to do so, you can go to File, Download, and you can download this tutorial as a Jupyter Notebook file. Before starting with the tutorial, you will have to do some setups. And at first, you will have to decide whether you want to run the tutorial on Colab or Jupyter Notebook. You need to decide this because based on your choice, you will have to perform different installations of libraries, and you will also access the data differently. So once you decide your platform, you will need to install necessary libraries. Now, if you decided to run it on Google Colab, the only library you need to install is NiLearn, and you can install it by running this command. Whereas if you run this tutorial locally on your computer, you will need to, prior to running it, you will need to have all these libraries installed. You can install them one by one manually, but we recommend you create an Anaconda environment, and then you install all the libraries in this environment. If you decide to do so, here we provide you with an environment file, which contains all of the libraries that you will need during the tutorial along with the corresponding version. And if you download this file, you can then open your terminal in the download directory and create an Anaconda environment running this command line, and then activate it through this other command line. If you do so, then all the libraries will be installed and you can go on with the tutorial. So once everything is installed, you will just import the libraries through this command. And next, you will have to access the data. So to access the data, you will first need to fill this form and accept the terms and conditions of the data. And once you do so, you will be provided with two folder links. These two folder links consist of two versions of the data, an unzipped version and a zipped version. You will use the unzipped version if you decide to run the tutorial on Google Colab. This is because since the data is unzipped, you can add a shortcut of it to your drive, and this will allow you to use this data to run the tutorial without having to upload any data at all on your Google Drive. So once you have this link, you just create a shortcut, and then you can easily run the tutorial. You will simply need to, after getting the link and adding the shortcut and mounting your drive, you will need to here edit these directories with the directory on your drive where you created the shortcut. This goes for the data directory, and then there will be a second directory you need to edit, which is the submission directory. And here you need to give a directory that the tutorial will use to save the predictions of your encoding models. If you decide to run the tutorial on Jupyter Notebook, then you will first need to download the zipped version of the data. This is because since the data is zipped, it will download faster. Once the data is downloaded, you will need to unzip it for each subject using this command on the terminal. And once you unzip the data of all subjects, similarly, you will have to edit these directories where you give the directory of the data that you're using, and another directory for the submission where the predictions of your encoding models will be saved. And finally, as a last step of preparation, you will have to select whether you want to run this tutorial on CPU or GPU. Now, the only part of the code which will be affected by this will be the image feature extraction by the AlexNet deep learning architecture. If you select GPU, this process will be faster, so your tutorial will also run in shorter time. However, if you don't have access to GPU, you can also run the code on CPU locally on your computer. It will work. It might take a bit longer, but it will still work. Once you're done with installing libraries, accessing the data, and selecting CPU or GPU, you will finally load and visualize the challenge data. At first, you will need to select the subject that you want to use during this tutorial. And here you have an option between all 8 subjects available in the challenge. And here, based on the subject of your choice, this code will define the directory in which the data is stored, and also create a folder on which the predicted data from your encoding models that you will later train will be saved. And next, you will load the fMRI training data. So the fMRI data for each subject consists of 2 files, one for the left hemisphere and one for the right hemisphere. And by the way, along with the data, you also have a README file that walks you through general data acquisition informations. It gives you the directory structure and files included in the dataset, along with an explanation of each of these files. Okay, once you load the fMRI training data, you will then print its shape, and you will see that for each hemisphere, left and right, the data consists of a 2-dimensional array, where the first dimension corresponds to the amount of training images, and the second dimension corresponds to the amount of vertices. Along with the data, we also provide you with regions of interest, ROI indices, that you can use to select the vertices belonging to specific fMRI ROIs, and then to plot these vertices on brane surface maps. So first of all, we're going to visualize all available vertices on a brane surface map. And here you can choose whether you want to visualize them you want to visualize the left or the right hemisphere. And once you select the hemisphere and you run this code, you will end up with this plot, which is interactive. You can move it, you can zoom in, zoom out, and this shows you all the vertices of your subject of interest of the left hemisphere that are used in the challenge. And next, we will be visualizing the vertices of one ROI of interest. Here is a list of different ROI classes. For example, we have early written topic visual regions, phase selective regions, and anatomical stream, and each class has different ROIs that can be indexed and visualized. So in the code below, again, you choose the hemisphere you want to visualize, and here you will be able to choose also the specific ROIs ROI that you want to visualize. We selected EBA in this tutorial, which corresponds to exostriate body area. And again, you have the visualization of it. So here, this is EBA. These are the vertices of the left hemisphere, subject 1 of the ROI EBA. And next, you will load also the stimulus images. So we provide you both the training and the test stimulus images. And once you load them, or better, once you access them, here you will print how many training images there are for your chosen subject and how many test images. And note that every image file has 2 indices in its name. The first index starts with either train or test, which tells you whether this specific image corresponds to the training split or the test split of the challenge. And then it is followed by a number. This number, or better, this indexing starts from 1, and it allows you to match the image to the corresponding brain response, fMRI response. So for example, this is image 1 of the training set. It means that if we go up to where we loaded the training data, if you remember, the first index of the training data corresponded to the training images, and that image would correspond to the first component of this dimension. Okay, so that's the first index, and the second index always starts with NSD, Natural Scenes Dataset, and this index starts from 0, and it allows you and corresponds to the 73,000 NSD image IDs. And you can use this to map the images back to the original file provided by the NSD dataset, and from there to the COCO dataset to extract image metadata such as segmentations, captioning, or the name of object categories present in the images. However, note that the images in NSD have been cropped from the original COCO dataset images. So if you want to use the original metadata, you will also need to adjust it for this cropping. And here we provide a link to a Jupyter notebook in a GitHub repository that you can use to perform this adjustment. And finally, the last visualization we provide is the visualization of brain fMRI response to a given image of interest. So here you can choose the training image that you want to visualize. Training because the test fMRI responses are not available. So you choose the training image, the number of the image. You again choose the hemisphere you want to visualize. And once you run the code, this is the first image 1, that we chose for the tutorial, and this is the corresponding brain response. And similarly, we also provide you code for visualizing individual ROIs instead of the entire brain. So this is the response to the same image, but instead of visualizing all vertices, we visualize the response of exostriate body area. Here you will train and evaluate linearizing encoding models using a pre-trained AlexNet architecture. And the encoding step involves 4 parts, where first you will split the dataset into training, validation, and test partitions. You will then extract and downsample image features using a pre-trained AlexNet architecture. You will linearly map these image features to fMRI responses, effectively predicting, that is encoding, the fMRI data. And finally, you will evaluate and visualize these encoding models' predictions. So first you will split the data into training, validation, and test partitions. Note that the training and validation partitions, they both come from the training split, the official training split of the challenge. So they both consist of both images and the corresponding fMRI responses. And in this tutorial, you will be using the training data to train your encoding models and the validation partition to evaluate them against the ground truth fMRI responses. Whereas the test partition used in this tutorial corresponds to the official test partition of the challenge, and as such it only includes test images, whereas the corresponding fMRI responses are not released. And here you will use the test partition to predict the corresponding fMRI visual responses and to prepare these responses for submission to the challenge website. So here you are, based on a random seed, selecting which training images will end up in the training partition and which of the training image conditions will end up in the validation partition. And specifically, the code is written so that 90% of the data, of the training data, will end up being training data. So this is the amount of training image conditions, and the remaining 10% will go into the validation partition. Of course, you can change this and you can use the proportions you want. Once you created these partitions, you will first apply them to the images. So for the images, you will create data loaders, which are classes, PyTorch classes, that are used to pre-process and load data prior to feeding them to deep learning models. So here you will create data loaders for the training, validation, and test images. And you will also define pre-processing pipeline. So these functions, they will resize your images, convert them to tensor, and normalize them. You then create dataset class and data loader class. And you see that each partition, train, validation, and test, has its own data loader. And again, you will be using this to extract, load, preprocess, and feed the images to the deep learning network, AlexNet. You'll then apply the same training and validation partition indices to split the fMRI data. So you will take the training data, the training fMRI data that you downloaded it, and apply these indices, and you will divide it into training and validation. Okay, once you've done this, once the data is ready, you will load the model, the AlexNet model that you will use to extract the image features. Once you load it, the model will be printed here. So since we're using AlexNet, you see that the AlexNet is can be seen as divided into two parts. One is the features and one is the classifier, where the features correspond to the convolutional layers and the classifier corresponds to fully connected layers. And here you will be asked which of the model layers you want to use to extract the image features. So we selected features 0.2, and if we go back to the model, you will see that features is here, 2 is here. So this means that we will select features from the max pool layer of the first convolutional block. Okay, so once the model is selected and you selected the layer you want to use, next in this function, you will define the dimensionality reduction algorithm that you will use, in this case BCA. And specifically we're using this because the image features of deep learning models are really extremely high dimensional. And to decrease computational load, we downsample them using, in this case, principal component analysis to only 100 features. So instead of having hundreds of thousands of features, potentially, it ends up having only 100 features, the 100 features which retain most of the variance. And next, with this line, you will, or better in this loop, you will, here you will loop through your data loader across the images, across image batches, and at each batch you will feed the image to the deep learning model. You will extract its features, and you will fit your PCA function. So now you're running the actual function, and this is the step that requires most time. I was running it on Google Colab with GPU, and it took me 41 minutes. And once you do this, PCA is fit, and now you can apply it to the data. And to do this, because of memory constraints in Colab, you will have to run the loop again and apply the learned PCA on the data, on the images. And this step doesn't take too long, it should only take a few minutes. And after this, you will end up with the downsampled versions of the image features extracted by the AlexNet architecture. So here you have the training images, training image features. This is the amount of training images. And as you can see, the second dimension is the amount of features corresponding to 100. Then you have validation, and then you have test. So once you have the image features, you will need to linearly map them onto fMRI responses, effectively encoding, predicting the fMRI data from images. And this is the encoding, linearizing encoding algorithms that we're using. So in the training step, as we just did, we were taking the training images, feeding them to AlexNet architecture to extract their features. And after downsampling them, we were using them to predict brain responses to those same images through a linear regression. So once the linear regression is trained using the training data, we apply it to the test data. So again, we take the test images, feed them to AlexNet architecture, extract their features, and then we will multiply the features, the extracted features, with the learned weights from the linear regression. And by doing so, we will predict the fMRI responses to these test images. And this is exactly what these lines of code are doing here. So at first, we are training the model. As you can see, we're using the trained data for both the left and right hemispheres. And once the linear regression, the encoding model linear regression is trained, we applied the trained regression to predict the fMRI responses to the validation images and also to the test images, using the downsampled AlexNet feature maps of these validation and test images. And this gives us predictions. So here we end up with predicted brain responses. And finally, we want to evaluate these responses. We want to know how accurate these encoding models were. And specifically, we do it on the validation data, validation partition, because we have, or you have, the corresponding fMRI visual responses, since they were coming from the original training partition. And we assess the encoding accuracy by comparing by looking at how similar the predicted data, the predicted fMRI validation data, is to the corresponding ground truth data using a Pearson's correlation, and we do so for both left and right hemispheres. And finally, we visualize these correlations, and we visualize these correlations using two different plots. First, we visualize each vertex correlation, so each vertex encoding accuracy on a surface, on a brain surface. And here you can see that vertices with dark colors, like black colors, they have zero correlation, so the encoding model was not performing well on these vertices, whereas vertices colored in yellow corresponds to correlations of 0.5 or higher, so our encoding model worked better at predicting these vertices. And besides the surface plot, we also plot the encoding results for individual ROIs. So here each bar corresponds to the average correlation of all vertices of one ROI. And this is informative if you're interested in knowing whether your encoding model is predicting certain ROIs better than others. For example, here you can see that the encoding models that we trained is better in predicting early ROIs such as V1. So in this final section, you will prepare the predicted brain responses to the test images in the right format for submission to Colab. Colab is the platform that we're using to collect your submissions, to evaluate them, and to calculate the evaluation metric from them by comparing them with the run-through fMRI test data. Furthermore, Colab also provides a leaderboard which is updated at every new submission, so you know how good your encoding models are with compared to the other challenge participants. It's important that before you submit the data, you convert it to float32 data type. And this is because Colab has a maximum submission file size. And if you keep your data at float64, this size will be exceeded. So make sure you convert it to float32 using this code. And once you do so, you can save your fMRI predictions to the test images using this code here. I highly recommend you to watch this video that walks you through the Colab competition website layout. It walks you through an example submission, and it also explains the submission outputs. It is also really useful because it will tell you exactly how you should format your data files and data directory structure in order to avoid errors during the submission process. And specifically, you should make sure that the subject directories have this name, that the data is separately provided for left hemispheres and right hemispheres, exactly with these file names, that these file names, or better, that these files contain the responses, the fMRI responses of all vertices of only the test images. And also you should make sure that when you submit your data, that you submit the data of all its subjects. And otherwise, if you do not comply to these things, the submission will result in an error. So once again, just watch this video. It will show you how to make a submission in the right way. And yes, so the submitted data should be zipped. So you will send a zipped folder, and here are some terminal commands to zip your data once you have the predictions of all subjects, predictions to the test images of all subjects. And finally, here there are some links you might find useful related to the challenge. And if you use any of the challenge data, please cite these two papers. I hope you will have fun during this challenge, and good luck.